All right. Gosh, this today took a longer time to get all sorted out. All right. How's it going? I'm ready. Seems like there's so little of you today, other than Mary and Maddie. Who are we missing? Where's Alejandra? Oh, huh? So should we give the +10 today?
Yeah.
They're so mean. Very, very mean. No, I will hold it for another day.
Aw. When we're not here.
When Emma's not here, remind me.
That day already passed. You're out of luck.
Oh my goodness. Okay. All right. So can you give me the basis or an example where enzymes are used for carbohydrate analysis? What's the basis of using enzymes for these carbohydrate analysis? Okay, let me show, yeah. Chop, chop, chop.
Cut the ball.
Very good. We cut the bond. That's absolutely right. So we use enzymes to determine the components, some carbohydrate components. So the carbohydrates will be the substrate to these enzymes. And one big example where enzymes play a major role, a big role, is in starch analysis. Because you first want to chop everything into singular glucose units. So you use a combination of enzymes. And then once you chop off everything using amylase and glucoamylases, alpha amylases and glucoamylases, then you have the glucose. Then you do also a coupled enzymatic reaction to determine glucose spectrophotometry. So this is key. And there are a lot of enzyme kits, so it makes it really, really easy because you can buy the enzyme kit if you want to do total starch, if you want to determine fructose enzymatically, or lactose, or sucrose. So there are a lot of commercial kits that are very cheap, easy to follow, and they give you really good sensitive results. So that question kind of answered, I answered both questions for starch. We use alpha amylase, not peptide, endoenzyme. So it breaks glycosidic bonds all over. And then glucoamylase is an exo enzyme that chops one glucose unit at a time. And then we use the coupled reaction. The first enzyme, glucose oxidase, with oxygen, you get peroxide. You use the peroxide to oxidize a dye in the presence of peroxidase, and then you get oxidized dye that is colored. Okay, so moving on to the dietary fiber. Yes.
Carbohydrates and vitamins very much not like
Total carbohydrate. Yeah.
methods of analysis, like using enzymes, initially used
for dietary fiber.
Oh.
So because, on the nutrition label, you have total carbohydrates, which is by subtraction. Then you have dietary fiber, which using the enzymatic methods.
The enzyme method is
for dietary fiber, yes. And then you use total sugars, and then you can use enzymes to determine total sugar and total added sugar. So yes, starch, you would determine starch for different reasons, not necessarily for the nutrition label.
And special methods are
yeah, I mean, an enzyme, the method used for starch is official method. Yeah. But not necessarily, you don't need it on the label. Okay, so dietary fiber, the simplest definition for it. It's the edible part of our food that cannot be digested. So we eat it, it goes in through our digestive system. It has different health impacts, but it's not digested and the components are not absorbed into our body or into the bloodstream. So, but it has different health benefits, soluble fibers, bulking agent, help with bowel movements, insoluble fiber with have impact on the microbiota. So we have different benefits for the dietary fiber. And that's why because of their different benefits and associated with prevention of some the cancers like colon cancer. So a lot of benefits, so that's why they're important and they're part of the nutrition label. So the dietary fiber, like you said last time, it's not only carbohydrate based. So they can have the lignin, which is not a carbohydrate base, but it's also present in plant sources. So that it's not digested, so it's part of the dietary fiber. Resistant starch is not digested, and it's part of the insoluble dietary fiber. So it doesn't have to be one class. The dietary fiber is not always your cellulose, your hemicellulose, your pectin. It might contain other components. All right, so soluble fiber, insoluble fiber, because they have different benefits, we need to determine the percentage of each. So it's not only total dietary fiber that is important for certain health claims. Sometimes you need the soluble fiber content versus the insoluble fiber content. So that's why we're going to talk about methods that determine total dietary fiber, soluble and insoluble. So your most common soluble fiber is pectin, but there are other hydrocolloids and gums. And also in oats, what's the most prevalent soluble fiber in oats that you hear about with health benefits? If you eat oats, you get a lot of beta glucans. Have you heard of beta glucans? Okay, well, it might come across later on in life, but it is a soluble fiber from oats specifically. Soluble fibers, they are the material from cell wall. So lignin is insoluble, cellulose is insoluble. Sometimes hemicelluloses are big group. So some of them are soluble, some of them are not. Resistant starch is considered insoluble dietary fiber. Whole wheat bran or whole grains in general are rich in insoluble fiber. But because of this really diverse components, they all have different monomers, they have different branching structure or even the chain structure. So is it possible to analyze dietary fiber components all in a single approach? No. So we can get a total dietary fiber, we can get total soluble and total insoluble, but we need different method to determine what are the components within the soluble fiber and within the insoluble. Long ago, and it's no longer in the book. It was in like the last time it showed up in the book was in the 3rd edition. And then ever since it was removed, the crude fiber. But I kept it because some of you might go out and work for pet food company. So pet food, usually they determine crude fiber rather than the dietary fiber. So crude fiber is determined by a method that is acid alkaline base, acid or alkaline, and sometimes using detergent. We used to have in this department a unit for measuring crude fiber, but it was I don't know where it went. It's no longer. It's not used anymore. It's just a unit. It's a reflux unit where you have hot plates where you put the flask with your solution and the acid and the detergent, and then you have a condenser, and then there will be a reflux for a couple of hours of digesting those fiber components. But it only determines certain components, like mostly lignin and cellulose, sometimes hemicelluloses, but it won't give you pectin or other hydrocolloids, um, other soluble fibers. So it won't necessarily give you the entire total dietary fiber. So that is why it's not used for the nutrition label. Because for animal feed, it didn't matter. The soluble fiber doesn't matter. So, okay. So now we're going to talk about how do we determine total dietary fiber, insoluble and soluble. It's a very tedious and long method of analysis. That's why we're not going to have a lab on it, but I would like you to really know the steps, and there will be an exercise of calculation that I will offer 5 points extra to turn in, what's today, Friday? How about Wednesday, next Wednesday? Because I know next Friday you'll have a lab report too. So we'll say by next Wednesday. The reason I say it's beneficial, you may or may not have a question about it in the final. You may or may not. So try to solve it, get those extra 5 points. Yeah. All right, so it's an important method. It is on the nutrition label, but given the time constraint, we don't have a lab. But I really want you to know it because so far we analyze almost everything on the nutrition label, and that's one important component. With that said, all ears, eyes with me on this complex method. All right, so we're after dietary fiber. So we just said dietary fiber is the component of food that cannot be digested. So this method digests everything except the dietary fiber using enzymes. So you start off, you want to digest the starch. What the point of digesting those other components is just to make them into their monomers that will be soluble and can be separated. All right, so large starch molecules, you want to get rid of them by using alpha amylase. But first, you want to heat and gelatinize the starch. Remember, in order for alpha amylase or even other sources of amylases to function and digest the protein, I'm just all fixated on protein. It's terrible. Okay, for all these enzymes to digest the starch, it has to be gelatinized. So we heat to gelatinize the starch, then we'll put alpha amylase and we allow it at certain temperature and time combination to ensure digestion. Then we change the pH for protease that we use to break down all the proteins into amino acids, or most of the proteins into very small peptides and amino acids. Then we add glucoamylase because you might have some small oligosaccharides or short chain maltodextrin. So you really want to make sure that they're all converted to glucose. So you add this glucoamylase and you allow all of these to digest, have enough time. So the kit, there's also a kit for dietary fiber, and it tells you pH, temperature, time combination at each step. So you have that all. Once everything is digested adequately, you can, if you just want to determine total dietary fiber, the total dietary fiber is composed of both soluble and insoluble. So in the soluble fraction, you have all of your glucose and amino acid and salts and everything that is soluble, plus the soluble fiber. So if you have pectin, beta-glucan, other types of fiber that are soluble, hydrocolloid, they will all be soluble with the glucose and amino acid. So, and then the insoluble dietary fiber will not be soluble. You can see them floating around or precipitating. If you add ethanol, then the soluble fiber will precipitate as well. So the ethanol will precipitate the soluble fiber. So now you will have soluble and insoluble precipitate. You wash, you filter, then you have your whole total dietary fiber, which is the filtrate, basically, that remained on the filter paper. So that's your total dietary fiber. Now, you might have some salt associated with it. You didn't wash them completely. You might have a little bit of small peptides that did not stay in the soluble fraction. So what we do is we take this total dietary fiber fraction that was on the filter, and then we measure ash content and protein content so that we correct for protein and ash, residual protein, residual ash. So we get the ash content, we get the protein content, we subtract it from the weight, and that would be your total diet. Questions? Yes.
So you have mostly glucose because you added the different numbers.
So you have now at this stage here, C, you have glucose, you have amino acids because you digested the starches, you digested the proteins, and then what's left is the small sugars and the glucose that came from the starch and the amino acid that came from the protein. They're all soluble components. But you also have a soluble component, which is a soluble fiber. You have the insoluble, which is the dietary fiber. If you want to get total, which is soluble and insoluble, you add ethanol. That will precipitate the soluble fiber. The insoluble fiber is already not soluble. So when you precipitate the soluble fiber on top of when you precipitate the soluble fiber on top of the insoluble fiber and remove the solubles by filtering, you get a fraction that potentially has a little bit residual protein and a little bit residual ash that didn't get washed very well.
That's the soluble.
Yeah, but you get most of it out. So some might bind slightly with your fiber and stay, but very little amount. That you add ethanol, you wash, you dry, but you might still have residual salts, residue basically that stayed on top of the fiber. So in order to get the correct percentage of total dietary fiber, you measure protein and ash in this fraction that you drive and subtract that from the mass. Questions?
Why don't you do the protein the first certificate? That way
But okay, it will be too complex of assays
Oh.
to do, and then you have starch. It will precipitate with the fiber if you don't digest it into small sugars. And the large proteins, it's kind of just like it's a lot of protein that you don't want in that total dietary fiber. Some of the protein will be soluble, some will not be soluble, so it will be a hot mess. So it's better to get rid of most of your macromolecules out.
So that's just what you do for the total dietary fiber, like you can't take that number and then split it into solubility.
We haven't gotten to this fraction. Okay. So this way, if you don't care about quantitating soluble and insoluble, you just want the total dietary fiber. So you can do it that way. If you want to quantitate soluble and insoluble, here's what you would do. So at this stage, C, where you have your soluble fraction and insoluble fraction, you will filter and wash. On the filter paper, insoluble fiber will be sitting. And then here, with the solubles fraction that went through the filter, you might have your soluble fiber. So the residue on the paper will be your insoluble fiber. Here you didn't put ethanol. So when you filter without the ethanol, all the solubles will go through the filter, and what's left on the filter is your insoluble fiber. So that insoluble fiber you wash, you dry, and then you take the weight and then determine the residual protein and ash, and you calculate insoluble fiber. Now, the soluble fraction that went through the filter, so the filtrate and washing, so you have the soluble fiber with the glucose, with the amino acid, with the other salts and solubles, they're all together. So you take that and then you add ethanol. Get rid of your small molecular weight solubles, and then you get your precipitated soluble fiber, you wash, you dry, and then you determine ash and protein, and then you get the soluble fiber. And now in this process, you got the insoluble fiber, you got the soluble fiber. If you add them together, you'll get total soluble. Question? Yes.
So would you specifically use this method, like the second part of the method, for when you just want to separate insoluble and soluble fiber?
And get
like
the quantity, yeah.
On the nutrition label?
Not just on the nutrition. If you just want to see how much of your fiber is soluble and insoluble for research purposes, for health claims, for all of that, then you would separate. Yeah.
And would you use like just the total dietary fiber method without separating the soluble and insoluble if you were just trying to determine like the full content? You don't
Yeah.
need to know.
Yeah, you don't care. You just want the total dietary fiber.
What is the more common method? The second one or the first one?
Well, it depends on the purpose. If your label has total dietary fiber and not split, then you don't have to do that. But if you're having a claim for beta glucan or soluble fiber claim, then you want to go and separate. But not only that, if you're doing research, huh? But I'm saying it's not only that. Also, if you're doing any research related. On the label, if you're making a claim, yeah, because I think the label is only total dietary fiber. It doesn't separate them into soluble and insoluble. If you're making a claim, then yes. But most of the time, we do the soluble and insoluble if we are, carrying a health study. We need to know if you are comparing, for example, you work in Georgia's lab, Kernza versus hard red wheat. Which one has more soluble? Which one has more insoluble? For health differentiation. Yeah. Okay. Other questions? All right. So this method is when you really want to determine the composition of your soluble fiber and insoluble fiber. So you want to know how much cellulose you have, how much pectin you have, how much hemicellulose you have. Then you need to look at the composition of the building blocks. So pectin, have galactouronic acid, cellulose have glucose, but you might have different branching and different organizations. So you can determine the composition of these fibers by doing the same process. You gelatinize, digest the starch, also hydrolyze the protein, and then the non-digestible polysaccharides and other dietary fiber will be hydrolyzed by sulfuric acid to liberate monosaccharides. So if all the neutral acids, that mean not the galactouronic acids, so all neutral acid, we can determine their composition using GCF-ID. And uronic acid in particular, we follow a specific method for it because uronic acid parts of the pectin by calorimetric detection. Speaking of pectin, usually pectin is determined for labeling of jams and jellies. They don't have any other insoluble fiber in there. So, and the only soluble fiber usually is pectin, used as a jelly. So, the way you do it is you precipitate with alcohol. And then you get the units of galacturonic acid and glycosidic linkages. They are very hard to hydrolyze with acid. So you saponify with a base so that you can you can remove the methoxy groups. So because some of these galacturonic acid are methylated, I'm hoping that you remember some of that from your food chemistry. I can go into all. Did you cover fiber with chem or not yet? Yeah. Well, pectin has methyl groups. Some of the galacturonic acid are methylated. So the first step is you saponify, remove the methyl. It's deesterification, like you did with your fatty acid. You deesterified from the glycerol backbone. So here you just deesterify the methyl groups and all of your units will be not methylated anymore. Then what you can do, then you can acidify and precipitate. So all of these carboxyl groups will be protonated and you precipitate with calcium pectate. Okay, so here's the calculation exercise. So if you were doing this in the lab and you took it through measuring insoluble and soluble, you will be collecting data. So I'm giving you your data points. So you will have the sample, you will run this in duplicates. So you will have the sample weight around 1 gram. And then to measure the insoluble and soluble, okay, you want to think of your process, right? So you need a container that will allow you to filter. So you will have some sort of a filtering unit. So when you get to the stage of separating the insoluble from the soluble in that method that I talked about, so you put your sample through that unit to filter to get the soluble pass through, and the insoluble will be on the top of that container, remains on that filter. Okay, so now you have your solubles. You collect your solubles in a different crucible and add ethanol so you can precipitate it. So you will have the crucible that has filter embedded in it, and then you take the weight of that. So you will have 2 replications, right? You have 2 samples. So you have the empty crucible weight for the insoluble, and the soluble that you will precipitate with ethanol and filter, you will have 2 other crucibles for them. Okay, so these are the given. You take the weight of the empty crucibles before you filter into them your samples. And then you have after you put in the sample to filter, you will have the insoluble fiber on that as a residue. After you wash and dry, then you take the weight. So the weight of that crucible plus residue, if you subtract the crucible, you get a specific weight. And then I'm telling you that part of it will have this much grams protein. And then the ash, you need to get the ash. So I gave you the weight of the crucible. Here, you subtract this weight of the crucible from that, you get the ash. Are you following? Did I lose you somewhere?
Are the values for protein and ash for both samples?
Yes, because we only have 2 reps. So 1 rep you would do protein on it because it's destructive, and 1 rep you would do ashing for it. So you get the ash content after you subtract the crucible weight. And this is the protein given to you by, let's say, Calda. Okay, so now you have the weight of protein, you get the weight of the ash, and the difference, so you take this 31.7235, you subtract 31.6372, you subtract the protein, and also you subtract the weight of the ash after you take into account the weight of the crucible. Then that would be the weight of insoluble fiber. And you do the same thing because the soluble fiber, you precipitated it with ethanol and then filter it, same thing. We always run a blank because the reagents that we use might be contaminated. So might be a source of protein contamination or salt contamination, ash contamination. So we always run crucibles empty, not having any sample or fiber from the sample. Okay, so the exercise would be average things out. Get the average sample weight, get the insoluble fractions, rep 1, rep 2, and get the average for sample and for blank. And then you continue with those, and they are self-explanatory. Do that exercise and bring in your paper for +5 points. Yes.
If we do it wrong, we still get the points for trying.
Yes. You still get the points for trying, and I will post the key. So I might not return your exact paper, but you'll see the key. Keep a copy of your paper. Take a picture. Wednesday.
What home Wednesday? Is it Friday? Oh, it's Friday.
It's Friday. Has it been such a short week? Yeah. All right. So next week we will determine total carbohydrate and total carbohydrate in lab using the phenol sulfuric acid method. Very simple but dangerous. You work with H2SO4 concentrated and a toxic phenol. So, but it'll be fun. And you'll have the beer and the 7-Up, but you won't be drinking. No. Don't get in. No, yeah, to analyze. Don't get excited. We're not going to hand out beer.
No.
Okay. So I have 10 minutes, so I'll start. A very interesting, another best topic ever.
Protein, you better not include them in. Oh.
What? What do you mean?
No.
That is not an attitude. Okay. Proteins are the best. Protein separation and characterization. So I separate proteins, isolate them, we characterize them. So here, this topic will talk about different ways where we separate proteins from different components.
Is this part of proximate analysis still?
No, done. New section. We're done with proximate.
Was the fiber?
Yeah, well, no. Total carbohydrate is part of proximate. The fiber is characterization of a component in the total carbohydrate. Yeah. No, protein separation and characterization has nothing to do with approximate analysis. It has applications in industry and it's relevant to research. Application in industry, why do we care about protein separation? Since you guys asked the question, is that proximate analysis? No, it's not. Why do we care about protein separation to go out to industry and work for cardio?
Stability of a product.
Huh?
stability of a product or how it behaves.
Yeah, one, but what is Cargill? Is it a company that produces what? Ingredient company. So what protein ingredients are there that you know? Yeah, egg protein, huh? Whey protein. No, concentrate, used as an ingredient. Pea protein, soy protein, egg protein, casein, whey protein, gluten protein. There's so many ingredients, protein ingredients that are used in a lot of different products. High protein beverages. If you're an athlete, you go and get a high protein beverage after an exercise, regenerate your muscles. Or high protein bar, just because you want more protein in your diet. Or a cancer patient, what is the Ensure? High protein. They tell cancer patients on chemo, drink more protein or eat more protein. So some of these ingredients go in there. Texturized vegetable protein, so you produce the ingredient. In order to produce the ingredient, you need to separate it from multiple components in the source and then purify, isolate, purify. So that's why it's important in industry. But there are other needs in research. We also separate proteins to characterize them, understand their behavior, figure out processing impacts on them. So the characterization bit, like you said, Scarlett, determining stability of the protein and its function, its nutrition during processing, for example. So these two aspects are important for industry, important for research. Okay, now how do we separate protein? It's based on differences in solubility. Just a? Yeah. No, all of them.
Gotten there.
Yeah, so we separate protein based on their solubility under different conditions. Some proteins are more soluble in alkaline conditions. Some proteins are more soluble in the presence of a little bit of salt. Some proteins are soluble in different concentration of acid and so on. So we can separate proteins like that. Solubility. And then adsorptivity, we can use chromatography to separate protein. So we can use adsorption chromatography to separate protein. Now you're going to say, is this applicable in industry? You say no. Who says no? We don't use chromatography in industry for ingredient production. You don't want to say.
Because it's expensive.
You say no?
I say no.
Who knows of an example was chromatography?
Yeah, wait, no, I take that back.
That was
take it back. I didn't.
What?
It's one to raise my hand.
She did chromatography for her.
Where?
They do it in this.
Where?
Microphones.
Yeah, but who knows of Agripur? It used to be the Visco. Do you know this?
Oh.
But Agripur produced whey protein isolate using ion exchange chromatography. And they had patents for a patent for the longest time. Now it expired. Nobody in the state were allowed to produce this ingredient using ion exchange chromatography, but the Visco and now Agripur, but the patent expired.
Isn't that?
Well, whey protein isolate is the most expensive protein ingredient out there. So it is more expensive than membrane filtration, and all other whey protein isolate in the country, like Glanbia, for example, they produce whey protein isolate by membrane filtration, is cheaper. But
It's not as pure.
it's cheaper, it has good purity, but just a sec. But the one that is produced by chromatography is much more functional. Higher functionality, so higher premium, more expensive.
I'm just curious about like if they use ion exchange chromatography in like larger scale, like
They
How
are.
big would the
Oh,
car?
very big. I didn't go to Agrippa, I didn't see, but you want to imagine like silos, like big.
It's like literally big.
Yeah, big column, like you see that HPLC column, magnitudes of size bigger, and they still doing media. And yeah,
Let's continue the other team.
but they're trying. So trust me, if they don't make profit, they would have gone out of business, but they didn't. So yeah, it's done. And chromatography is applied not just for protein isolation, for other like pharmaceutical applications, separation of different ingredients, production of antibodies. You know that antibodies are produced to produce ELISA kits, you know, the enzyme link immunosorbent assay. So that you would find antibodies purified by chromatography. So yeah, so it is chromatography is used in industry. It's just more expensive, so anything that comes out of it will be more expensive. Okay, I just went on a tangent. Denaturation, yes, because you can heat proteins. Some proteins are more heat liable. That means they are susceptible to denaturation more than others. So you can separate by heating. Some proteins will stay soluble, other proteins will precipitate. Okay, not going too far apparently. But let's answer this one.
Oh, no. All of them?
What's that?
All of them.
No, you were gonna say something else.
I said A through C.
A through C?
Yeah.
Oh, another creative way of all of the above. Okay, so it is all of the above, but listen, this is very important. And students make that mistake when answering questions on the exam. So you can use electrophoresis to separate protein on a gel, but you cannot produce an ingredient using electrophoresis. So you cannot separate and collect, to purify and collect. You can separate them and observe them, qualitatively look at differences. Whereas size exclusion and salting out, you can actually collect a protein, purify a protein. You can separate by all of them. You can do size exclusion and look at the chromatogram and separate protein based on size. Very good. You don't have to collect. You can salt out, like in a hydrophobic interaction chromatography, and look at chromatogram. You don't collect. But electrophoresis, you separate and you don't collect and you will not collect. So did you get that differentiation? So not every time you separate the protein, your target is to collect. Okay, if your target is to collect the protein fraction, you don't use electrophoresis. Just remember that. It will be helpful to remember this. All right, you guys, we'll see you on Monday. Have a great weekend. Enjoy the sun. It's pretty cool outside.
Nice weekend.
Thank you.
You're welcome. I'll see some of you at college school.
I can make it today.
No.
I have a quiz.
What time?
College ball.
4.
4.
Middle to the where?
153.
Here.
Come,
Pop in.
hop in. Come, Alan. I dare you.
Let's see.
It was fun.
It was fun.
Yeah.
Seriously, it's fun. It's a game.
4 hours.
4 to 5. You can leave in half an hour. Just come check it out. You might not want to leave us forever.